Good morning, good afternoon, good evening to everyone, and welcome to the World Federation of Hemophilia Virtual Summit. This is the beginning of 5 days of medical and multidisciplinary as well as professional sessions for our global hemophilia community. It's my pleasure this morning to introduce the medical session, the medical plenary, with Steve Pipe and Luigi Naldini. Steve will lead off and discuss current ongoing AAV clinical trials and what to expect. And then Luigi will come in and talk about novel therapies such as lentiviral gene therapy. Steve is a professor of pediatrics and pathology at the University of Michigan, where he's been for a number of years. He did all of his training, most of his training in Canada, and then came down to Michigan to lead the hemophilia program as well as the special coagulation laboratory. Steve has done a tremendous amount of work both in research as well as clinical studies in hemophilia and bleeding disorders, and is currently the chair of MASAC, Medical and Scientific Advisory Council for the National Hemophilia Foundation. It's my pleasure to ask Steve to get started and tell us about AAV gene therapy. Steve.
Well, thank you, Glenn. Thank you to the WFH organizers and also to the Aracenius Fund for the honor of presenting this plenary lecture on delivering on the promise of gene therapy for hemophilia. These are my disclosures, and I do want to point out that I have provided consultation for a number of gene therapy programs and also serve on several steering committees. So what is the promise of gene therapy? I would like to offer that this has the potential to be a single treatment, so-called one and done, that leads to a lifetime of steady state expression of factor 8 or factor 9, that would be sufficient to liberate patients from any concern of recurrent hemophilic bleeding, if not even normalized coagulation. They would be liberated from the need for continued prophylaxis, even for the need for factor replacement following trauma or prior to surgery. This kind of health equity would have been unimaginable several decades ago. This quote from the famous inventor Alexander Graham Bell highlights that although we stand on, I think, the threshold of this achievement, delivering on this promise is going to require broad-based multi-stakeholder preparation from scientists, manufacturers, federal regulators, health technology assessors, persons with hemophilia, national advocacy groups, and multidisciplinary healthcare teams. And they're all going to have to be focused around an emphasis on delivering education, on ensuring that we are approving safe and effective therapies, that we remove any barriers to access to these therapies, and that we demonstrate excellence in the clinical delivery of these therapies. Harold Varmus, the former director of the National Institutes of Health, published an editorial in the New England Journal of Medicine shortly after the completion of the Human Genome Project that really has ushered in the genomic era of medicine. And he outlined what he described as 4 troubling questions that would need to be addressed to realize the potential of genetic therapies. These include, how can we better train the next generation of physicians to practice genetic medicine? How can increasingly complex genetic knowledge be made readily accessible to all practitioners when they need it? How much will the expanded use of gene-based methods further escalate the cost of healthcare, and who will pay for it? And how can we ensure that these products of our science, largely financed by federal dollars, will actually reach the citizens of our country? I think these are relevant questions today, even though these were penned about 18 years ago. And I'm certainly going to be addressing issues around education and access this morning. What an incredible series of advances we've been witness to in the past few decades, but really accelerating in these last few years. The replacement therapy era didn't really begin until the late 60s and early 70s with plasma-derived clotting factor concentrates. Hemophilia was one of the earliest treatment areas to benefit from the recombinant DNA era. And that technology has been leveraged even further as targeted engineering has further improved the biochemical properties of these molecules. We're only a couple of years now into new non-replacement therapy era with a first-in-class humanized bispecific antibody, emicizumab, which substitutes for factor VIII replacement. There are other antagonists of the natural physiologic anticoagulants that remain under investigation, seeking to hopefully rebalance the hemostatic system. But now we are about to enter the gene therapy era. Now, gene therapy includes things like gene editing and cellular therapies, but the first clinically available technology for hemophilia, at least, will be through gene addition. This graph, I think, nicely characterizes these therapies according to their impact on measurable factor levels across the bottom portion of the graph and the clinical correlate of effective blood clotting or hemostasis across the top. Now, beginning on the left, traditional prophylaxis is characterized by the peaks and troughs of factor levels, which are then mirrored by a similar effect on hemostasis across the top. The extended half-life agents really maintain this same paradigm with the advantage of less frequent delivery and perhaps even higher trough levels. The non-factor therapies, we no longer have a measurable factor level, but clinically patients are able to realize maybe a subnormal correction of hemostasis, yet with the advantage of steady-state clinical effect. With gene therapy, we have the ability to have a measurable steady state level, perhaps even fully in the non-hemophilia range, that's going to be matched by a steady state clinical hemostatic protection. Now, unfortunately, the builds did not come through in this presentation, so I'll have to walk you through what I wanted you to see today. You know, if we're going to call this a genetic revolution, we should remember some of the fears and prejudices that accompanied previous genetic revolutions. Public perception initially of so-called test tube babies spawned all kinds of wild speculation about the genetic age. People had visions of babies growing inside test tubes, farms of genetically engineered babies. Now, we know this to be a simple yet elegant procedure of in vitro fertilization and subsequent implantation. But, you know, many of those fears were not quelled until they saw the fruits of the science. And this is Elizabeth Carr, who was the first test tube baby, if you like. And once people saw this, I think it changed the public prejudice considerably. And we now know that millions of families have been helped by this technology. Now, what I wanted you to see here is the world is quickly being introduced to lives who've been transformed by genetic therapies. I was going to show you a picture of a little girl who otherwise would have a devastating progressive disorder of spinal muscular atrophy, who's received gene therapy to arrest any progression of that disease. A boy with a devastating degenerative brain disease who received gene therapy that's going to stop the progression of that disease. CAR T-cell therapy is now essentially routine in the treatment of high-risk bone marrow and blood cancers. And then this boy on center picture has an inherited blindness and received a intraretinal injection of gene therapy that's going to stave off, hopefully, any progression for him for blindness. So I think what people are missing in this genetic revolution hemophilia is many of us have not met people who've gone through this treatment and been transformed by this therapy. So we need to have at least a basic understanding of what this first wave of gene therapies for hemophilia are going to look like. These are going to be an adeno-associated virus-mediated liver-directed approach in which a functional copy of factor VIII or factor IX is packaged inside a viral vector, the AAV vector. That vector has been stripped of its propagating viral gene elements, and then following a single outpatient IV infusion over as little as 1 to 3 hours, these particles are going to be picked up by liver receptors, taken up into the cell, where the vector particle is going to uncoat. It's going to deliver the DNA to the nucleus of the cell, and there the genetic elements that accompany the gene are going to allow for efficient expression and secretion of factor VIII or factor IX into the plasma. Those proteins are going to achieve a steady state balance between the rate of secretion coming out of the cell and the rate of clearance that normally would occur for those factors. And that's going to be the steady state factor level that we'll be able to measure with traditional blood sampling. Now, there's lots of hurdles here that have to be overcome. Many people will have encountered versions of these non-pathogenic viruses previously, and they may have specific antibodies that would otherwise block the uptake into the liver. For 25 to 40% of patients, they will actually be excluded from this therapy on account of this. The immune system also gets one more crack at responding to this viral vector, as after uncoating within the cell, the protein elements are going to be broken down and they're going to be presented on the surface of the cell to the adaptive immune cells. And if these cells react to what would be seen as foreign protein, then an immune response that could actually destroy the cell could ensue, taking with it any potential factor expression from that cell. This has actually been manifest in the trials by elevations in common liver enzymes, the so-called liver toxicity of AAV gene therapy. And this really must be diligently followed post-infusion for weeks and even months afterward. Despite these hurdles, the studies are actually going to show that this works in the great majority of individuals, and that the immunosuppressive course that would be required to suppress or dampen down this adaptive immune response can actually be avoided in some cases or be short-lived for many. So the first approved therapy is likely to be here in August, which I'll tell you about shortly. Now, our focus today is on what I would like to call a pathway to preparedness for getting ready to deliver gene therapy. I think through our hemophilia treatment centers. This will involve education, continued evaluation of safety and efficacy, ensuring that there's access for as many patients as possible, that our sites are properly prepared and have overcome any hurdles at their individual sites, and ultimately all culminating in excellence in clinical delivery. So let's start with the educational challenges. The ISTH conducted a survey of healthcare teams and scientists, over 5,000 respondents. 66% were physicians, 59% were directly involved in the care of patients with hemophilia, and 35% of them indicated that they'd lacked the ability to explain AAV gene therapy for hemophilia. 40% indicated that they had limited ability or a lack of comfort in answering patient questions about gene therapy for hemophilia. This is from a WFH survey that covered 76 countries, 103 associated with NMOs, 109 physicians. 68% of the persons with hemophilia reported only a basic understanding of gene therapy, and 44% of the treaters indicated only a basic or intermediate understanding of gene therapy. So I think these are significant challenges. I found this publication from Dr. Misbah and colleagues quite helpful, entitled How to Discuss Gene Therapy for Hemophilia, a Patient and Physician Perspective. They go over what is the rationale for gene therapy, what is it, how does it work, what safety and efficacy data support its use in hemophilia, how should we discuss this with patients, where do we go to get additional information. What this is about optimizing the lexicon or the language for effective communication. There's been other efforts. Last year at the ISTH, Dan Hart and some other colleagues presented a multidisciplinary effort on really capturing this lexicon and language. And I think you'll see more publications and more efforts in this regard. The other thing that I think is going to be key and that I've found helpful in talking to patients is how to share a meaningful educational story that they can engage with. So I want to share one example that I have liked to use. This comes from an effort with multiple colleagues that I hope will be published shortly. So this uses the analogy of a package for delivery. So we're trying to answer the questions for the patients. Well, what is the delivery package? So this is a viral vector. It is the delivery package that's designed to transport a good copy or a functional copy of the gene to its destination. In this case, it's a good copy of the clotting factor that's going to be delivered to the liver cells for patients with hemophilia. So what's actually in the package? Well, at the factory, if you like, where this is manufactured, that package is going to be filled with a functional copy of the gene, as well as materials that are necessary to switch on that gene to allow it for production of a functional clotting factor in the host. How's the package going to be delivered? Well, the delivery package, if you like, is stamped with an address so that it can reach the liver. This is so we can convey the principle of tropism that's conveyed by the vector coat, the capsid of the AAV vector. That package is delivered by injection. It travels through the blood to its destination. The liver recognizes it and brings it inside and unpacks it, just like we do packages every day. So what happens to the package upon delivery? Well, the contents of the packages are kept inside the liver cells. The packing materials are then recycled. This allows for a discussion about the potential for an immune response to those packaging box, if you like. The immune system does have an opportunity to recognize that packing materials as unnatural, and it could stop subsequent packages from being successfully delivered. So this allows us to talk about some of the immunologic consequences of gene therapy. So it seems that we have a lot of work to do collectively to ensure we're educated, our colleagues are educated, our patients are educated. And what I highlighted in the state-of-the-art program is a number of efforts which are, I think, available to you right now. The ISTH has a multi-year effort that's ongoing. It's up to date. It includes a review of the most recent data at meetings. It has active from the very basics of hemophilia all the way through some of the challenges that we have ahead. The WFH has its own effort on gene therapy for hemophilia. You can go that on their e-learning site on the website. The National Hemophilia Foundation, the European Hemophilia Consortium, these member organizations are also driving education and helping to hone the lexicon and the language of gene therapy. Medscape has a wonderful CME-based program that is updated regularly and has really excellent concepts about gene therapy, the clinical trial results, potential clinical applications. And we can even go to some of the larger general gene therapy societies like ASGCT, who also have sort of gene therapy 101 type efforts. I think all of these are things that we should be engaged in. And so if we're thinking about this first step in the pathway to preparedness, education is what we should all have been concentrating on right now. With regards to safety and efficacy, well, of course, this is going to come from the clinical trials. Now, who's actually been getting gene therapy? These are men who have been at least age 18 or older. They have severe hemophilia A or B. They've been well previously exposed to factor 8 or factor 9 products. They've all come into these late trials on a period of prophylaxis to compare traditional prophylaxis to gene therapy. To date, nobody with an inhibitor has been treated with gene therapy in these registration trials, and that includes historical inhibitors that go way back to childhood, even if they've been fully tolerized. From an exclusion perspective, I mentioned the challenge of pre-existing immunity to the AAV capsid, which will screen out a number of patients. Because we're targeting the liver, we have to have at least a well-functioning liver. Patients do not have to be negative for hepatitis B or hepatitis C, at least exposure, but they do have to have gone through proper eradication of hepatitis C and have to demonstrate that RNA is negative. HIV is not always an exclusion in these clinical trials. Now, what I've focused on here are the late phase 3 clinical trials. There will be more coming, but this is just to highlight the ones that we should expect most immediate approval. Nicely, we have 2 programs for hemophilia A and 2 programs for hemophilia B, which are close to completion. These are going to involve 2 different types of AAV serotypes. That's going to give us some options in the clinical delivery. 1 each for hemophilia A and B that are an AAV5 based capsid, and then a bioengineered capsid for both A and B as well. The Hem B programs are all using a hyperactive form of factor IX, which is going to ensure that we get the highest factor activity possible through these programs. And the phase 1 and 2 studies have been presented previously. I present the publications here. You're going to hear many updates at even this meeting and perhaps at ISTH coming up. And these are going to give us the insights about what we should be expecting, hopefully, to see from the phase 3 programs. Now, what should we be looking at? We should be looking at 2 key things: efficacy and safety. Under efficacy, there are the physiologic and clinical parameters. What's happening to bleeding events? What are the factor activity levels being achieved? What's the duration of expression? What about pain and discomfort in joints? There are mental health aspects to be focused on. How does this impact overall emotional functioning? With safety, there are short-term adverse events, long-term adverse events, and then, of course, on all platforms of therapies, we have to be concerned about mortality. You're going to hear a number of things. Glenn gave a wonderful introduction yesterday about some of these adverse events that are still going to be challenging. We've mentioned liver toxicity, but we want to be evaluating for inhibitor development, immune responses to the gene therapy. Is there any risk for thrombosis at any level of expression from these treatments? Are there any concerns about vector integration of the DNA into the host genome? How long is the duration of the vector neutralizing response after this therapy? So all of these answers are going to come from the phase 3 trials. The next thing we have to be concerned about is access. Incredibly, the FDA expects to receive 200 investigational new drug applications per year for gene and cell therapies by this year. And it expects to approve 10 to 20 of these per year by the year 2025. They've recognized this is going to need for major budget increases for the agency. I hope our country still has money left to do this. Streamlining across agencies to ensure that these get through the regulatory approval process. Manufacturers have to step up as well. There's going to be need for improved ability to scale manufacturing, maybe new technologies, new expertise, expanding capacity. Some of this is going to come through acquisitions and maybe strategic partnerships. Comparative effectiveness, I think, is going to be a key aspect of evaluating gene therapy, and it will help with the health technology assessment. The Core Heme project, which has been published recently, was a multi-stakeholder effort to identify a core set of outcome measures that have actually been incorporated into a number of the late-phase gene therapy trials. These are also going to help streamline the regulatory approval process. Institute for Clinical and Economic Review, or ICER, has indicated that they are conducting a comparative effectiveness review of BMN 270, the first hemophilia A program likely to be up for approval, that will directly compare it to traditional factor VIII replacement and emicizumab. So I mentioned that we are on the cusp of this. Company BioMarin has announced that they have a priority review with a review action date of August 21st, 2020. And there was an indication that there would be no advisory committee currently planned to review the application. This has the potential to be the first approved gene therapy in the U.S. For the treatment of any type for hemophilia. And they have fast-track status, if you like, in the EMA as well. So this is coming. But there's still going to be some other things to get over. Even after regulatory approval, we're going to have to deal with, well, how are we going to get access to this? What's the reimbursement going to look like? These are likely to involve innovative payment approaches. Many have suggested that the reimbursement may be linked to specific outcome measures. For instance, persistence of the factor activity, continued bleed control, reduced or eliminated need for factor replacement. So regulatory approval is really just step one. Then we're going to need a viable pathway for access, prior authorizations, drug acquisition, and reimbursement. So the next thing we're going to talk about is site preparedness. And a lot of this comes from what I've learned doing clinical trials at our center, but I'm now having to rethink and getting prepared for delivering clinical gene therapy at my center. Institutional preparedness for product handling and administration is really key. There are infection control committees that we had to get through. We had to get through issues around nursing handling of the product and how it would be delivered, patient and staff precautions when we're delivering gene therapy. Pharmacy preparedness is really key. Are they able to receive, handle, and store this material? Do they understand and have the capacity for the thaw time that's involved? Do they have the containment procedures for preparing these products? And one thing that I'm learning in going to the clinical phase is clinical pharmacy is not the same as research pharmacy. These are two different areas in my hospital, and I'm going to have to go through this process again with our clinical pharmacies. You have to know where you're going to administer this, where you're going to observe the patient after administration, how are you going through subject selection, and how are you going to do follow-up. And as with most things at our hemophilia treatment centers, familiarity, regular practice are all going to contribute to improved outcomes. So site preparedness is sort of the checklist, if you like, of what are the hurdles that we need to do at our center to overcome so that we can actually be prepared for clinical delivery. So the final piece is how do we do real clinical excellence in gene therapy? Well, this is a statement that jointly from EHAD and EHC on this idea of a hub and spoke model, where basically the gene therapies would be prescribed and managed by an expert hemophilia comprehensive care center, perhaps even national hubs in some places, and then monitoring would occur at hemophilia treatment centers who would maintain close communication with the primary expert hub. This has actually been realized in many places. Even in the gene therapy trials, there were many centers like ours which served as a dosing hub, and afterwards the patients went back to the other study site for their follow-up. And the principle here is, even though we talk about this as a one-and-done therapy, it does not mean get and forget. There's going to be significant follow-up that's required to ensure that we get the best outcome for patients. So we should all be striving for excellence, and we want to ensure that we're delivering gene therapy and ensuring that we're delivering good outcomes. Now, I mentioned at the beginning that the perception of a genetic revolution is changed once people get to meet people who have undergone these therapies. I found these two gentlemen from Toronto who went through gene therapy recently, and notably, these were their comments about going through the gene therapy process. The individual on the left said it's pretty magical, and his brother indicated it's life-changing. And I think as we meet, and you're going to meet at this meeting people who've undergone gene therapy, and I think it will change all of our perception of what this treatment can mean for patients. So in closing, this was the Arrhenius lecture, this famous painter who also had hemophilia from Sweden. And I really like this picture that he painted entitled "The Button of Life." My youngest grandson recently discovered his belly button, and it's interesting to see how young children are drawn to this small portal from which their life sprung, and they want to show everybody. Our genes are the fountain from which springs all of life. And how exciting that hemophilia now can rediscover its own belly button as it enters into the genomic era. And we can't wait to show everyone what this is going to look like. So thank you again to WFH and to the Arrhenius Fund for the great honor of being able to give this lecture.
Thank you very much, Steve. Really appreciate your perspective on gene therapy and its development in hemophilia. It's my pleasure now to introduce Luigi Naldini. Luigi is the director of the San Rafael Telethon Institute of Gene Therapy in Milan, and has been working in this field for his entire career. He first began working with lentiviruses back in the mid-1990s and has done really much of the seminal work in developing a vector system out of lentiviruses, which is now progressing, and he has conducted a number of clinical trials in the area. He also has, over these past 10, 15 years, continued to look to develop lentiviruses for hemophilia. So, Luigi, we look forward to your talk, and we will do questions at the end of the talk if there is time. We have a number of questions now for Steve, so please continue to put questions into the Q&A section as you listen to the talk. Luigi.
Thank you, Glenn. Thank you, and good morning or good afternoon to the audience. I decide now to move on to discuss the potential use of a lentiviral vector for the gene therapy of hemophilia. Steve Pipe really nicely explained to you how hemophilia can provide a relevant therapeutic option to a hemophilia patient. And gene therapy, of course, is, as mentioned, can provide one-time dosing and stable expression of factor, which I think is a key advantage in terms of phenotypic correction of the disease. And as Steve Pipe showed you, there's a lot of effort towards clinical development of AAV-based gene therapy, which has proved safe and efficient in several early trials, and now in the next phase 3 trials, and soon, hopefully, on the market. So why do we need additional vectors or strategies? Well, essentially, there are a number of unmet needs that we are trying to address. One, which was also mentioned before, is the fact that a sizable fraction of individuals are pre-immune to AAV, because AAV is a common viruses to which most of us become exposed and immune. So these patients are not eligible, at least for standard AAV-based gene therapy. Maybe in future, engineered virus will do, but that's really a big unmet fraction of patients. And the second important aspect is that because AAV mostly remain extra chromosomal, so the cargo, the genetic cargo of the liver remains outside of the chromosome, if the cell divide and proliferate, they may lose this new genetic information. And so while it may work well in adult individuals, as has been shown in the clinic, it may fail to maintain stable expression in young individuals in which the liver continues to grow. And so that means pediatric patients are not currently eligible again. And these are the essential key features we are trying to address by using lentiviral vectors, because lentiviral vector, and this is really the rationale here, are essentially efficient integrating systems. So the virus has a machinery which can physically insert the genetic cargo into the chromosome of the infected cell. So even if the cell then will continue to divide, it will essentially transmit to the progeny the acquired genetic information. The other potential advantage is that HIV, of course, is relatively uncommon, luckily in the general population, and so there will be a very low prevalence of immunity to that virus, preventing use of the vector in those individuals. And finally, the HIV-derived vector also have a larger genetic cargo capacity as compared to AAV. And so complex transgene, especially factor VIII type of transgene, may be more effectively delivered to patients. So the design for delivering gene therapy through the lentiviral vector to the liver is essentially using this advanced generation lentiviral vector, which are now commonly used in the clinic in other disease settings, mostly in ex vivo gene therapy, as I will mention towards the end of my talk. And these are made, essentially, are hybrid particles which contain a fraction of the original HIV virus, not more than 15% of the genome and just 4 out of 9 of the viral protein to assemble the particle. This, of course, is a replication defective, and also use a different envelope than HIV to enter into the target cell. Now, importantly, for effective liver-based gene therapy, we need the vector to express essentially and only from the hepatocyte, which was achieved by a combination of hepatocyte-specific promoter inside the vector and also microRNA-mediated regulation. Besides the technicalities, what you see here is a picture of a mouse liver in which you see upon reconstruction that only cell which lights up with the GFP transgene after delivery of the vector are the big and round hepatocyte and not the other cell type. This is important to prevent also immune response to the vector. Now, over several years, as also Glenn mentioned, we have been testing these platforms in animal model of hemophilia, starting from mice and then dogs. We have a number of dogs treated both with early generation and more advanced vector type for several years now, up to 9 years for the first dog actually to be treated, reaching a stable level of factor over this long follow-up period and correction of the bleeding phenotype. More recently, we have tried to further engineer this vector for better performance when moving eventually to human. And one of the approach we took, and this is a little bit of virology, if you mind, the vector is really buds from the surface of a producer cell, which is a human cell. So the vector requires a human membrane, which is good in terms of essentially preventing sensing by the immune system. But in this membrane, there are also molecules which can be sensed as foreign. For instance, major histocompatibility complexes, which are different from many individuals to others. And so we have modified the producer cell, for instance, to remove any major histocompatibility complex so that the particle could not be sensed by at least these features. And also, we have increased the expression of a factor which is naturally present on cell, which prevents phagocytosis. And so this now makes the vector more resistant to clearance by innate system and more able to reach the hepatocyte when we deliver IV. And we have tested this new vector design in non-human primates, which is, of course, the ultimate model. These are not disease models, but, of course, they are relevant models before moving into clinical testing. And as you can see in this picture, we have had a very nice tolerance of the infusion, which was intravenous peripheral vein infusion. There has been almost no affected changes in the plasma chemistry, except for a very moderate elevation of enzyme, not even clinical. And we got substantial level of factor 9 expression. This is also a hyperactive factor 9 that you see here. And what you see in the yellow dot is actually we even reach supranormal level, up to 300%. So this is not what we want to reach in humans, but the good news was that the vector performed even better in monkeys here than in dogs and mice. And this is most likely because the vector has been derived from a human virus. So it's best apt to infect human cell, and the surface, as mentioned, also has human feature. And so it may best interact with human or non-human primate system. So the advantage, of course, is for once we could now scale down the doses of vector, and what you see in the yellow arrow on the bottom is recent data in which we could give a lower dose of vector and reach a stable expression at about 30% of normal, which could be for factor 9 a good initial target for testing. The monkeys which receive and express very high dose of factor eventually also develop antibody to the factor. This has been observed also in other cases and platforms, and is mostly attributed to overexpression of the human factor in monkeys. This was not seen for factor 9 when we use the lower doses. And importantly, as shown here, we measure the presence of the vector in different organs of the monkeys at the end of the experiment, and the vector was essentially only found in liver and spleen, showing that this is a remarkably liver-selective strategy, not only in expression, but also at the infection site. So the biodistribution is favorable, and as shown here, this recapitulates this benefit that we found when moving in terms of the effective dose of vector to reach about 30% stable factor 9 activity in the blood. You can see that while in mice we need about 3 times 10 to the 10 vector particle per kilogram, this was slightly lower in dogs, and essentially it was almost 1 log lower in non-human primates because of these features that I said best interact with humans. And this is at the moment is the predicted potential effective dose in humans as well, when we eventually move to clinical testing. Now, as I mentioned, one of the challenges in the field for gene therapy still is to stably and long-term express factor VIII because of the larger complexity of the gene as compared to factor 9, including the size and also the site of expression. So that was an important goal for us. Can we use this platform to stably express factor VIII as well? As you can see here on the left, these are 3 different transgene versions for factor VIII that we administer to the lentiviral vector platform into mice. And you can see that what type of engineering we can do to the genetic sequence to improve expression. To the left, you see essentially a B-domain deleted wild-type sequence, which expresses to a few % of the norm. When we optimize the codon users, so to make the genetic sequence better translated in the vector, we reach more than 100%, even in this case up to 700% expression level. And so this was just made that sequence more efficient in translation. A further modification was also to make the protein itself more stable by addition of XTEN, which is a non-structured polypeptide, which protects in part this protein as well as other proteins in circulation. And this also, the recombinant factor VIII coupled to this XTEN moiety is also being safely being tested in humans as well. And so we can now, by this improvement in the design, once again establish, as you can see on the right, stable expression of factor VIII, even administered in newborn mice, and then we can follow through the life of the mice, and the factor remains stable. So essentially showing the prediction that we can deliver the vector into a highly proliferating tissue. And of course, once again, we will be able to scale down doses when we move to a larger animal, as you see here in non-human primate again, which receive different doses of this factor VIII XTEN transgene through the lentiviral platform, and you see we are within the target range of reconstitution around from 30 to 60% of stable activity. Now, as commonly observed here, for factor VIII, we do see essentially all the monkeys to develop antibody to the human transgene. This is usually attributed to this highly immunogenic nature of this molecule and the human source. And again, this has been seen with other platforms, including with the recombinant factor, and then is not seen in humans when you move to at least pre-treated patients. So hopefully that will be the case. We think this observation is in line with other ones. But of course, we are carefully at assessing immunogenicity in this case as well as for all maneuver. So I think I'll show you we can reach a therapeutic target for factor 9 and factor 8 in non-human primate by a single administered dose IV, which is well tolerated. We are also trying to further improve, enforce the rationale, to further strengthen the rationale of our strategy by trying to learn more about the mechanism of gene transfer and stability of the vector. One of the questions is to make sure that there is lifelong and stable expression. Which liver cell are we reaching? As you know, during liver growth, not all hepatocytes contribute to the same extent to liver growth, and in particular, there is like liver damage. Again, the growth may be driven by stem cell-like or progenitor-like hepatocytes rather than the majority of the mature ones. So we are evaluating the maintenance of the vector upon different conditions: the growth, steady state, and damage as well. And just briefly, you can see, first of all, in this slide that you see while the liver is growing after newborn injection of this GFP delivery vector, you see the clusters of GFP-positive hepatocytes which grow and expand, clearly indicating the proliferating hepatocyte maintain expression and give rise to larger clusters of cells. This is in a normal proliferation condition where hepatocyte may contribute. As I said, we are looking what a different population will do. At the moment, we don't see any difference here. And interestingly, we also look at so-called liver stem cells, if you want, which are assayed by the capacity to generate organoid in vitro. These are usually ductal progenitors, which can be isolated from liver and give rise to this differentiated epithelial organoid. And after we administered the vector to newborn mice, and then we isolated the liver, and we grew in collaboration with Hutch and in the UK. We grew up this organoid. You can see that some of them, a fraction around 10% of them, were vector positive, indicating that we could reach also stem cell-like cell in the liver. So guaranteeing that even presumably we are testing that in a disease setting, the vector would still be maintained in the liver long term. And in terms of the other aspect which is relevant to this platform, which is integration, of course, this is a two-edged sword. The advantage is stable expression and maintenance throughout potentially lifelong stability. However, integration into the chromosome is also potentially harmful because the vector can integrate semi-randomly throughout the genome, and occasionally insertion of the vector may disrupt an essential gene or may alter the control of that gene and possibly give rise to an expanded clone of cell, which potentially could become malignant. Malignancy has not been seen in humans treated with lentiviral vector gene transfer. We have several hundred of patients treated both with hematopoietic stem cell and T-cell gene therapy using lentiviral vector. And in particular, hematopoietic stem cells are known to be very sensitive to transformation. They were transformed by early generation integrating vector, but this has not been seen with lentiviral vector, indicating an improved safety. And of course, this is something we really value, and we are stringently tested in the liver. We have tested lentiviral vector platform into tumor-prone mice from liver delivery, so that we could have an oversensitized system. And here again, we have not seen increased tumorigenesis by the vector platform. So that overall would provide us with substantial confidence that this vector system may provide not only efficacy, but also substantial safety. I think I show you a favorable translational profile of this HIV-derived vector because of the human fitness, essentially, of the original virus. They behave efficiently in non-human primates and in human cells in vitro and ex vivo. This vector, as I said, they are being widely used in human hematopoietic stem cell, and they have been shown to be highly efficient. And for liver-directed gene therapy, we are shown up to a non-human primate feasibility, highly well tolerated to IV administration, stable and robust factor 9 and factor 8 expression. There has been antibody development for the high doses in the monkeys. However, recapitulate what has been seen in other platforms. And so overall, this vector may eventually, when they hit the clinic, address outstanding needs of the field, both in hemophilia and considerably other disease, which would require early administration of the therapeutic gene and stable long-term expression. With that, I'd like to acknowledge the people, both in my lab, in particular Alessio Cantore, who is the leader of this project for long term, the other people in my lab that are shown here, together with our collaboration with Bioverative and now Sanofi, which has been sponsoring at least the good part of the later study in the monkeys, and is committed to develop this eventually for clinical use. And with that, thank you. I'll be happy to take questions.
All right. Well, thank you very much, Luigi. Am I online? Hello? I'm not hearing my voice. And can we get Luigi and Steve back on? Okay, terrific. Thank you both for wonderful talks to lead off this morning. We have a lot of questions, so the only way we can get through a number of them is if we get short answers from the two of you, okay? So I'll go back and forth between you. Could lentiviral vector be the solution for other rare bleeding disorders, especially due to the size of the proteins? Luigi?
Well, I think it depends on the target cell. I think if you do IV administration, hepatocyte and endothelial cell can be transduced quite efficiently, and we can engineer expression selectively from either of these populations. So they may be potential use for that as well. I think it's a platform of version. We haven't studied all beyond the factor 8 and factor 9. So I just say potentially yes.
Okay, thank you. And then how can you control the location of the chromosome where the gene is integrated? So that's a question for Luigi, but also would apply to AAV as well, where there is a small amount of integration that occurs. Luigi?
So maybe I start, Glenn. So yeah, that's a great question. Unfortunately, we cannot. That would be wonderful, and it's one of the major issue of these both lenti and early generation vectors. The integration is semi-randomly, so each cell receives an integration at a different place. Most of the chromatin will tolerate the insertion. Remember, we only have less than 1%, which is coding, and also if you include regulatory region, that still makes a lot of space for insertion. So most of the insertion will be well tolerated. But in case that insertion occurs next to like a cancer gene or a proto-oncogene, it may be harmful. The vector, however, is designed to limit those adverse interactions.
Okay, thank you.
And respect to AAV, I was just saying, respect to AAV, related to your question, AAV does not require integration into the chromosomes to be functional. In fact, the DNA tends to circularize primarily, remains what we call episomal inside the nucleus. This is also the detriment for AAV, because if the cells divide, the episomal DNA does not get replicated. However, because we're giving trillions plus of particles to the liver, even a small percentage of integration events, whether that's 0.1% or 0.01%, I don't think we know exactly. We know that genetic material can end up integrated into the chromosomes. It does not appear that there has been a demonstration of what would be called genotoxicity related to those integration events. They appear to be somewhat random, if you like, but do not seem to have a predilection to those hotspots that Luigi had mentioned.
Thank you, Steve. So there's a number of questions about the question of antibodies to AAV, and maybe I can just dispense with this. Question about corona and adenovirus. All of these viruses are completely different from one another, and antibodies to AAV does not mean antibodies to coronavirus or adenovirus. They're all completely separate and independent of one another. Is there any issue in terms of the capacity to produce vector? So I think I'd like to ask you both that question. Steve, do you want to start with the AAV?
Well, I mean, I think there could be, but I would think that the manufacturers are trying to be on top of this. You know, there's been a limited number of facilities available worldwide for gene therapy. And I mentioned 200 INDs per year coming to the FDA. A lot of them are using the same types of manufacturing facilities to prepare product. The larger manufacturers, I'm sure, have their own capacity, but they have to ramp those up because preparing product for 10 patients is not the same as preparing product for 1,000 patients. So almost certainly capacity expansion is going to be a key part of this next phase of preparedness.
And Glenn, I can essentially underwrite the same answer.
Vectors.
The manufacturing this vector require, of course, a complex process. It's quite similar in reality for HAV vector and AAV-derived vectors. You use transfection of plasmid, from cell you harvest, you purify. This has been satisfactory to go up to phase 3 trials now. Commercial development, which is ongoing, also for lentiviral vector, will enable further this platform. So I think this may be a bottleneck in terms of timing, but it's not a hurdle which we'll not be able to overcome soon.
All right, thank you. And then another question that can apply to both of you. What's the time based on the clinical trials, and then Luigi based perhaps on the primate studies? Time frame for steady state.
Okay, I might have missed part of your question, Glenn, but you know, from the point of delivery, which you know, this is a single treatment event, infusion over about 1 to 3 hours. However, expression can be seen within days and even a couple of weeks. Because of the availability of long-acting factors now, a patient takes a single long-acting dose right before their gene therapy infusion, and that's potentially the last dose of factor that they will require. The expression level kicks in quickly enough to cover them before that factor wears off. The peak steady state probably is not achieved until around 12 to 16 weeks.
I may have missed the initial question. Of course, I understand. I guess from what was the previous answer. So it's probably very similar in terms of administration. Is IV
Luigi?
infusion. Yes, can you hear me? Glenn, I may have missed your question, but in terms of IV infusion. Yes, can you hear?
Yes.
So steady state level.
I may have missed your question, but sorry, I'm still on. I think I got confused here.
So Luigi, I wondered about
steady state levels for lentivirus.
Sorry, there's a problem with noise here. I think we reached the steady state level in around 1 week, so it's quite fast. I think it's faster than AAV because of the mechanism of integration after the infusion, and the infusion is similar.
Okay, thank you. A question about B domain deleted products, and perhaps for both of you. Steve, is there any increased risk of inhibitor formation when using B domain deleted products, which is what are being used in all of the AAV clinical trials?
Yeah, and practically this is the case because the size of the viral capsid can only accommodate a truncated version of factor VIII, and the B domain deleted is the one that has been chosen for all platforms to this point. Unfortunately, we don't have a hyperactive form of factor VIII, at least that anyone can envision right now. So, but remember, all of the patients coming into these trials are well experienced. They've been on factor VIII exposure for usually many, many years. There has not been any evidence of triggering an immune response to factor VIII itself through gene therapy delivery.
Okay, and Luigi, are you working with full-length or B-domain deleted factor VIII?
We also work with B-domain deleted, although we don't have those strict limitations that AAV experimenters need. I must say that most of these transgenes are being tested as recombinant factor, also safely in human. However, the concern of immunogenicity is right, not so much because of in gene therapy we may change the transgene. I think it's because we deliver the gene sequence together with the virus, and the virus may potentially, of course, activate the immune system at least temporarily. And this could sensitize to immunization. So that's why there has been a concern in the field for long term that we would not incur in that issues. This luckily has not been seen until now. Of course, as mentioned, the patient being treated until now were previously exposed to factor replacement, and so they were tolerant to anergizing, and there has been no breakage of that tolerance, which is the good news until now. But we have to remain carefully, I think, attentive to that.
All right, thank you, Luigi. We are going to need to end, unfortunately. I wish we could continue, but I would like to very much